All right, let's get started. I believe I answered the question about C-peptide cleavage. Go ahead and ask if you did, if I did not. Come on, mouse. Okay, can everyone see my PowerPoint? Someone say yes, if so. No. Share. Better? Chat. Good. Okay, so we'll go through regulation and gene expression. Before I do that, let me show you, I just added this little link here on the bottom left. This is something I set up for students a few years ago because they were interested in, like, how do I become the doctor I want to be? So if you're interested, go to this. It's just a resource I made if you want or not. Long story short, you type in your name here, or a fake name, so no one knows who you are. And then in columns B through C, or B through I, you enter in your information, and you can put this in over time. How many research experiences you had, volunteer, work experiences, publications, what's your COMLEX level 1 and 2 score. And as you do that, it will, in real time, color code how competitive you are. So green is good, red is bad. I've converted everything to one number. There's a README that tells you how I did this. And over here on the right, you'll see per discipline how competitive you are in basically standard deviations. So in red, for example, not to kill anyone, but who has some information. So like put a name 1, row 21. God, if my mouse can work, I'll try to highlight it, but I can't. My mouse isn't working. It looks like, whatever, I can't highlight. Okay, I can't move my mouse. But anyway, you get the idea. So like, long story short, some disciplines are just much harder than others. They're much more competitive. So like dermatology, for example, you see a big red column. You need a ton of publications in that one. I show you where I got the data, so have at it if you want. Okay, and all those sorts of things, you can email me or tell the liaisons if you have questions, and I'll pass them on. So you have this lecture and then 2 hours Friday epigenetics with me, synchronous, and that's the last one for synchronous lecture for me. By far my favorite lecture that I give you guys is the 2-hour epigenetic one. So attend if that's your thing. It's a fun lecture, I think. It's very applicable. Anyway, but this lecture is kind of like a preface to that, in that it's about gene regulation, which epigenetics is, but we'll get into a lot of the more applicable examples there, okay? So we're going through some of the molecular machinery here. All right, again, a lot of objectives that are very discrete. See, they're all like 1, 2 slides, 1, 2, or 3 slides. All right, again, you'll see boards because there's a lot of board testable stuff. So how does aspirin work, which is also called salicylate? It basically blocks I-kappa beta, I-kappa beta kinase, which normally I-kappa beta kinase activates NF-kappa beta, which increases inflammation. So that's one mechanism that aspirin works. So on the right here, I'm showing that pathway. Here's TNF. It binds the TNF receptor. Then that activates I-kappa beta kinase, which causes I-kappa beta to be phosphorylated. That phosphorylation causes it to be polyubiquitinated, which now we understand that means that blue polyubiquitinated blue thing I'm showing here, that gets degraded, and that releases this teal, I'll say, the teal NF-kappa beta to go into the nucleus and do its thing. So aspirin blocks this step, okay? So I-kappa beta is kind of like a anchor holding NF-kappa beta from doing its thing, okay? And when I kappa beta gets phosphorylated, it gets polyubiquitinated and destroyed, and then NF kappa beta can go on and cause inflammation. Well, aspirin keeps that anchor of I kappa beta because I kappa beta kinase can't phosphorylate I kappa beta. So we've gone through DNA to RNA to protein to modification. Now let's talk about how you're controlling if those happen or not. So from the DNA step, you can obviously control transcriptional regulation. I'll talk a lot about that in the epigenetics because that's what that is, turning on or off DNA. Once you have RNA, you can control the processing. We've talked about alternate splicing, right? Then you have mRNA, you can have, does it have a poly A tail or not? Does it get, put out here into the cytosol? Once out here, you can control if the mRNA is translated. Does the poly A tail get degraded so the mRNA is degraded? Once you have the protein, you have that post-translational modification we talked about that can inactivate or activate it. Okay. I won't go through this. This is the same slide you saw before, the RNAs. So polymerase I, there's rRNA genes. There's hundreds of tandem repeats, and there's a large pre-rRNA. So it gets processed into these 28S, 18S, and 5.8S. So there's one promoter, right? This binds, and then you make the 18S, 5.8S, and 28S. Can anyone tell me why this is a good evolutionary design to have them all together under one promoter? Type a C so I can call on you or just start talking. Okay, Joseph?
Does it keep them proportional in amount?
That's exactly right. The stoichiometrically equivalence will always be correct, right? There's no way, there's no reason you would ever want two 18S's for every 28S. It would be a waste and they'd be floating around. Who knows? I don't think that would be a deleterious effect, but you always want one, one, and one. Okay, so there's no reason to ever have a different promoter and upregulate one or the other. Okay, so you have this promoter that's upstream of the gene. General transcription factors bind, UBF and SL1. Okay, so you have polymerase and these two other things that bind. Polymerase II, right? Those are coding for the mRNAs. Polymerase II for us needs transcription factors, and a lot of our genome is set up for transcription factors, 5%. General GTFs and gene-specific GTFs, and so now we're going to get into those for eukaryotes. So you have your poly 2 promoter sequence, okay, TATA, and it's about 25 to 30 base pairs upstream. Now, it is called a cis element. Students get confused about this. A cis element is, imagine a DNA strand as this thread, okay, so from one end of the chromosome all the way to the other is one strand, right? If you bind to if the gene is on this one strand, if you could go on that strand and go left or right and go along that thread and there's a sequence that affects that gene, that is a cis element. Cis meaning close, right? It's on that same strand. A trans element would be something that's coded on another strand that then comes in and affects this one. So oftentimes those are going to be transcription factors, or something's made from another sequence on a different strand, and then some proteins coming and binding and affecting this protein. That's an in trans, okay? So this TATA sequence binding is a cis element, and the binding order, I color-coded these words to match this picture, and it goes from the top left down to the bottom right. First, you have TF2D, then TF2B, RNA, then this big purple RNA polymerase 2 comes in, then TF2F, H, and then, TF2F, then TF2H and E. So you can follow it all here. TF2H has helicase activity to start to unwind stuff, right? And it also has kinase activity. I will point out some of the lectures have been shuffled. So TF2H has these components called XPB and XPD. In prior years, I would have already taught you about them, but I think Dr. Gardner is teaching you about them later now, in maybe year 2 or in the spring. Long story short, these XPs are called, are termed for a disease, Zema pigmentosa, and then the enzyme BD or whatever. And long story short, those can have, if you have a deficiency in those subunits, they're involved in fixing DNA mutations. And so people that have this can't be exposed to the sun because they have massive lesions. And so those subunits are used here, but here those subunits are involved in their helicase activity. Okay, and so there's this C-terminal domain of TF2H that has this, normally this nucleotide excision repair, right? That's the XPB and XPD. So this purple dotted thing that you see, that is the C-terminal domain, okay? And so when it gets long enough, it bends back on itself, and then it gets phosphorylated, and then the elongation process can continue, okay? And so this is part of that protein, right? This C-terminal domain that's shown in circles here, they're just showing a continuation. And down here, you see in blue, they're showing that RNA being made. Okay. And you saw a mediator, which we had talked about before. Okay, so for polymerase type 3, it is unique in that it binds the promoter region, and then all the other machinery that binds downstream of the polymerase 3. And so this isn't how I would have designed stuff. You know, it's counterintuitive because the polymerase in this picture is going to bind and want to go to the right. And you have all this stuff in the way. Okay, so it's different in that regard. In addition, I've circled kind of the things to know here. It has that TATA binding protein, TBP, but that TBP it's shown up here because it's not binding the TATA region. And so this is an example of how things evolve and change over time. The TATA binding protein evolved to bind the TATA region and also have other parts of it that interacted with the other proteins and machinery. And then for polymerase 3, it's using those other aspects to interact with the machinery, but not the portion that's needed for binding the promoter region. All right, so let's talk about response elements. The cis elements I've already talked about are those DNA sequences. Promoters, the general transcription factors bind to the promoters. Far from them in promoters, there are enhancers and silencers, okay? Enhancers often activator transcription factor proteins that bind, and silencers are often repressors, right? I'll talk about like how those are named, right? When we discover them, we name them for one thing, and they could bind somewhere else and do something else. But normally you'll be taught, okay, this is an enhancer or a silencer if it's important. And those can change in cancer with mutations, and we'll talk about that. Okay, so those are pretty self-explanatory, co-activators, co-repressors. Insulators are a little harder concept to grasp. So imagine, I don't know if you can see I'm using my hands. I don't know if you can see me, but you can look in the bottom right too to see this. If you have a gene here and a gene here, okay, and which way do I want to show this? Let's say that, I'll show it in your direction. Let's say that the promoter's here and you're going to go up and make this gene on my one hand. Something's binding here, and maybe that's going to bind, and let's say it's a HAT, a histone acetyltransferase. So it will acetylate the histones and loosen up DNA, right? So it can get activated. Well, this is binding and acetylating a bunch of histones, right? That HAT. You don't want the HAT to come here and then acetylate a bunch of stuff over here and turn on a gene that's nearby. So a silencer, if this is the HAT coming in, a silencer would come in and bind the DNA and kind of block the HAT from interacting with other stuff. So a silencer could kind of be thought of as like a dampener of other things. okay? Sorry, that an insulator can be thought of that. That everything I just said was an insulator, right? It's insulating the effect. Okay, and so cancer has loss of function in some of these for enhancers, right? As you can imagine. And so if you have an enhancer, this is an example for a cancer. If you have an enhancer that does a good thing, like let's say an enhancer turns on P53, right? In cancer, if the enhancer is something that comes in and is a hat or does something, right? If it keeps the domain, I'll hit this again in another slide, but if it keeps the domain that binds the DNA, but it loses the domain because it's been mutated in cancer, if it loses the domain that actually opens the chromatin, that enhancer has now become a silencer because it binds where you need to bind to turn on the gene, but it blocks it. It's just blocking other stuff from binding. Hope that makes sense, and I'll have it in another slide. Right, so we've already said transcription factors can up or down genes, turn them on or off. And here's a big picture where you're seeing all this complex form, and then you see this big protein called mediator. And mediator has kind of evolved to be a hub of everything that needs to interact. This is a big protein that basically can interact with other proteins. And you see how this brown thing is the DNA, how the DNA can wrap around. So there can be an enhancer segment, there can be a cis element, right? Because this is on the same strand way over here, but it bends back. And so there's a protein interacting with it and then mediator to affect if that mRNA is going to be made, okay? There's also this gigantic loop protein called cohesin, which I find amazing that this is, it's this massive protein structure. Here's what the structure actually looks like. And so there's a hinge region, so it can open and close to kind of snap the DNA loop around so distal parts can work. And this is an electron micrograph where you can actually see that diamond shape because it's so big, okay? That's just an FYI, clearly. Okay, so another board thing again, here is, we're talking about turning on Cree genes, okay? So let's follow this pathway. I'm showing it in words up here and the pictures to the right showing it. CAMP comes in and binds this regulatory region, okay? And ends up increasing PKA and CREB. CREB goes in and binds and turns on Cree genes, okay? So CREB binds this Cree element down here and causes transcription, okay? So CREB plus a co-activator shown here in light green stimulate the expression of the gene, right? So CREB is coming in, binding something, and if another co-activator comes on, it can turn on transcription. So all this stuff works by, not all this stuff, but the stuff binding cis elements works by binding DNA. Here are 4 DNA binding domains you should know. If any protein has this, the protein binds DNA, okay, or evolved to. Zinc finger, helix turn helix, leucine zipper, and helix loop helix. And here's an example showing it bind DNA, and then there's a separate domain that's an activation domain, right? So things have evolved to eventually a DNA binding domain can, in one generation, swap and get added, and then suddenly there would be a new protein. I mean, that happened on an evolutionary time frame. Okay. And here, sorry, I'm showing the DNA binding and reaching out with the activation domain and touching mediator. That can happen like this, but remember, it can also loop out and be much farther away too. Okay, and so clearly the transcription factors, you don't need to know these consensus sequence, but the transcription factors have unique consensus sequences that they find, right? So CREB binds, for example, TGAC, TCA. You don't need to know that, but the point is, and P53 has its own, the point is that consensus sequence can be screened in the genome and it can bind multiple places, and we know, okay, here are the things that are up or down. And so if someone, I mean, you're not going to be mining databases to find this, but as a clinician, when a cancer patient has a mutation, in a transcription factor, you'll easily be able to know what pathways are down because of that or up. Okay. So I mentioned that one of the binding domains is zinc finger. So if you have a zinc deficiency, that can cause a decrease in transcription factor efficacy because they can't bind correctly without zinc. Zinc's involved in zinc deficiency also causes decrease in wound healing, adult hail, itchy blisters, hypogonadism, dyschezia, and anosmia. So this is taste being altered, so you're tasting different things. Anosmia is loss of smell, so that can be like that comes into play in old age homes where unique populations are kind of not consuming enough food because they have these that make food less desirable to them. And it can zinc deficiency also predisposes you to alcohol cirrhosis. So as an example, but this is not common, enteropathica A is an autosomal recessive disorder where you have this zinc channel defect. So you don't have it, it's lethal if uncaught, but if you know about it early, you can have lifelong zinc supplements, and then you're okay. It usually presents when a baby is weaned off of breast milk because they're getting the zinc from that. Okay. So here's an actual view of looking down a histone. So those colored ribbons are the histone, and wrapped around it is the DNA. And so we'll get again this in the epigenetic lecture, but you'll see this picture a bunch of times. I try to just keep this light bulb on so you can see it. So here is a lysine. Here is the HAT, histone acetyltransferase. It adds the acetyl group, and that gene is now on. And then histone deacetylase takes the acetyl group off, so the light bulb is off. The gene is off when it's deacetylated. These are very drug targetable. We have drugs now that are, for example, HDAC inhibitors, right? So you have an HDAC that would normally turn a gene off, right? It takes the acetyl group off, and the inhibitor keeps that gene on. And so much like, you know, I think in the future, chemotherapy, our current chemotherapy will be thought of as archaic because we're attacking all cells that divide. This is why you lose hair when you're on chemotherapy, but cancer divides a lot, so you're attacking those pathways. HDAC inhibitors, likewise, are not nearly as specific as we want, right? They're affecting a lot of genomes because the molecules we have are just not as specific, and they affect histones more generically than we would like. They're getting better. All right, so here in words, I'm showing you the pathway of the HAT and the HDAC. HAT shown on the bottom left, HDAC on the bottom right. So on the bottom left, a HAT, histone acetyltransferase, binds an activator, right? The activator bound some cis element, and then it had a topography that allowed the HAT to bind. The HAT binds the activator, acetylates histones, loosens the DNA, increases gene expression. Whereas an HDAC, histone deacetylase, on the right, bottom right, binds a repressor, deacetylates histones, tightens the DNA, stops gene expression. Okay? So now it should be clear that I talked about CREB, and CREB was a co-activator. It's a HAT. It is a histone acetyltransferase. Okay. And the enhancer element can an activator and a repressor can bind an enhancer element. Enhancer doesn't mean activator. Okay, it just means something that changes the expression of the gene. All right. So again, back to the SERMs, selective estrogen receptor modulators. I try to show you different figures showing the same thing to get this one tamoxifen thing because it's confusing. So here are the antagonists in breast. Tamoxifen is used to help cancer. In the uterus, raloxifene reduces endometrial cancer because all the green is agonist, all the red is antagonist. In the uterus, both tamoxifen is associated with endometrial cancer, so that's bad, it's in red. And in bone, raloxifene and tamoxifen have agonism, but raloxifene is better. So that's why raloxifene is used to help osteoporosis and tamoxifen is used for breast cancer. And here I'm showing you the enhancer element in bone up here. I'm showing the estrogen receptor bound and tamoxifen bound and a co-activator. And then in the breast, estrogen receptor bound, tamoxifen bound, and there's a co-repressor. Okay, so the estrogen receptor can bind the same enhancer element, the same DNA, in different tissue, but because there's different co-activators and co-repressors in that tissue, the binding of the estrogen receptor does different things. Okay, and then tamoxifen and raloxifene interact with the estrogen receptor differently enough to cause different effects with these co-activators and co-repressors, right? So that's the take home of how you're deciding those things. You've seen that slide again. That one, I know I showed you that exact slide and this exact slide. I don't know if I had this table in the last one. So here's the kind of summary take-home table, right? So tamoxifen, so first of all, you have to worry about your cancer patient. Are they ER positive? Are they ER estrogen receptor positive? So a patient that has breast cancer could be treated with tamoxifen if they have an estrogen receptor positive in that cancer cell line, right? Because then it will respond because tamoxifen will bind something. And then oftentimes what happens is they'll respond to treatment, but eventually the cancer will mutate away and become estrogen receptor negative. And tamoxifen will not work against a cancer patient that's ER negative because they don't have the target to hit, okay? So in ER positive patients, tamoxifen is used in premenopausal women. This other one, fulvestrant, can be used in postmenopausal women. And for osteoporosis, raloxifene is used for osteoporosis, okay? And that can kind of be seen from this, this chart over here to the left. Fulvestrant might change over time because more studies are being done with it. But you can see why, you should be able to look at this and see why, well, anyway. Okay. So moving on, here's an example where there's competitions for the enhancer site. So imagine that this green is an activator, okay? A repressor could have the exact same, I mentioned this before, the exact same domain as the activator, but just not have that top part. And if that's the case, it will compete and lower the activity of the activator because it's binding the cis element and the activator can't come in and bind. Okay? All right, so now the same thing. I'm trying to hit things again and again. Here's that same TNF that we saw before, and here's aspirin coming in and where it affects IK beta kinase. Well, corticosteroids, see the boards all over the place, very infinitesimal. Corticosteroids interact right here. So they're also inhibiting inflammation, but a kind of a step further down, okay? So I have the flow chart in words up to the top left, okay? And inhaled corticosteroids stop cytokines and decrease NF-kappa beta. And they're first-line therapy for chronic asthma. But anytime, one of the downsides, so inhaled corticosteroids can cause oral thrush, which is a development basically of bacteria growth. And so anytime you have a disease where you're having inflammation that's causing a problem and you're taking something to suppress that, you are basically lowering your immune system, right? You're saying like, don't inflame and you can have opportunistic infection because your body's not responding we're always exposed to bacteria, right? All right, so here on the bottom left is showing corticosteroid, which comes in and binds growth receptor and can go in and turn on gene activation and gene repression for different things, okay? Yeah. Okay, so in cancer, transcription factors are up and down, right? Because they do different things. And so it depends on what gene you're talking about. So for oncogenes, right, so those are genes that cancer likes. In cancers, there is a gain of function. And there's a big difference here where you only need one allele to be damaged for that oncogene to do something. So let's say like MDM2, right? MDM2, cancer likes MDM2 because it shuts off P53, right? So if one MDM2 were mutated to always be on, that's an oncogene causing an oncogenic effect because it turns off P53, right? Whereas for P53, you need a double hit. You would need both P53s inactivated. This goes back to my whole thing about, do you need to be homozygous or heterozygous for general things? If it's an enzyme, you need to lose both of them because half of P53 is enough, okay? And so here are examples. c-Myc is involved, a transcription factor that causes Burkitt lymphoma, MYC-L1 for lung tumor, and MYC-N for neuroblastoma, and all of those are oncogenes, so there's a gain of function, whereas WT1 is involved in Wilms tumor, and it's a tumor suppressor, so you need to lose both of them. I'm not going to try to draw this base. Oh, okay. I don't have a good drawing mechanism here. I'll try. Okay. So in short, there's this thing called the Warburg effect that's kind of been, it was a good story, but it's kind of been disproven why that's happening. But so you should know what's going on in cancer, and they're still working out exactly why that is. But follow on. Before I do that, let me get out of that. And here, for Panopto's sake, that should work. Yeah, here's a fuller chart, and I'll kind of just show some of that. But basically, the posit was that cancer cells are growing so fast they sometimes seem like they would be oxygen deprived. And so there's an increase in anaerobic metabolism, which is not ideal, right? If you're growing a lot, you want the most efficient way to produce energy, but cancer cells don't do that as much as you can measure that, and that's the Warburg effect. But then what's been shown is that cancer cells seem to do that regardless of if there's oxygen or not, and so it doesn't fit the exact story we wanted, but nevertheless, knowing kind of what pathways are happening still make things targetable. Okay, so the take home that I would get there is that, I don't know how to change colors, but color, let's make this red. So you have growth factors, okay? I don't know how that turned blue. Oh, okay, sorry. Growth factors. And they're gonna bind growth factor receptors, okay? And in cancer, that's gonna happen, and you don't really want that to happen. And then that's going to be on the outs, So here's the cell, and that is going to increase mTOR. mTOR, mammalian target of rapamycin, is what that stands for, is basically your detection of fed state, or one of them, right? That increases c-Myc. I'm trying to make this is all kind of red and bad, and that ends up going down to lactate. So lactate is this measure of this Warburg effect. And so the other way you can see this is you have glucose, and I've talked to you about, the glutes, so GLUT5 or whatever, or the SGLT1. Then anyway, glucose comes in, and then there's a bunch of arrows. You get pyruvate, and goes to lactate. Now, ideally, if you were growing, this pyruvate would go over. It's acetyl CoA, I'm running out of room. This is, and I have a bad mouse, such a bad mouse. That's not a crossed out acetyl-CoA, sorry. This is in the mitochondria. Basically, you end up with a lot of ATP, right? And that's good. You make a lot of energy when you have oxygen. All right. So I'd go with the slide I showed you on the other one. But that's the big take home. All right. Transcription factors are important everywhere. You've probably heard about these Hox genes involved in development. We share them in an evolutionary sense with, insects. If you look at an insect, kind of the segmentation of their body, different Hox genes are turned on to make each of those segments. It's kind of cool sometimes the amount that the genome kind of matches the 3D manifestation of how we end up. I mean, there's crazy examples of, like, in our brain, the neurons that control our fist are in the order of our fingers. So the thumb neurons there, anyway. For this Hox gene, if things go awry, you can end up with things like extra digits polydictyl, and that's predisposed in certain populations. So Indians are more often polydactyl than others. This is Indians in Asia, not Native American Indians. Again, an FYI, but here are a bunch of disorders that are associated with mediator. I said this mediator is this gigantic protein that's evolved to detect everything. Clearly, if that goes wrong, there are a lot of problems. There's actually probably less than I would have than you might think, because it's so important that I think probably oftentimes when something goes wrong with mediator, a fetus doesn't come to term, would be my guess. But there are plenty of examples of problems with mediator. All right, and then lastly, a lot of times when we have, I guess I might call it polymorphism or a mutation, it's not always on off. A lot of times it's more like a dial where you're augmenting the amount that it interacts. So not everything's going to be this whole thing's dead. This is a protein is interacting with this complex with mediator only 20% as much as normal, and so it's having less of a transcription and then downstream effect. All right. I believe that is it. Go ahead, let me know how that is, and any questions. You can type them in, type a Q, and you'll be out early today. All right, I will see you Friday, 2 hours of epigenetics.